And we are coming to work package 2, which is also or is the heart of the project, the product development. And I'm very happy that Stefanie Mittermaier from the Fraunhofer Institute for Process Engineering and Packaging in Weinstefan near Munich in Germany is here to present her work in Work Package 2. Stephanie is a food technologist and has a strong focus on plant food products. Yeah, Stephanie, please start your talk.
Thank you very much, Dorothee, for inviting me, and I hope you can see my presentation. Yes?
Yes.
Okay, great. So first of all, I want to thank also my colleague, Susanne Gola, for doing the work within the Appetite project. Unfortunately, she could not present today her own work, so I'm presenting it today on behalf of her on the development and characterization of innovative plant protein fiber products. And within the Appetite project, we had 3 different goals we wanted to achieve within the work package 2, which was the first goal was to have an optimal amino acid profile, and the second to determine all the different amino acids. And what you can see here is on the left-hand side here, you can see the FAO reference, which has an amino acid score of 1. And the next one in gray is the whey protein, which has an amino acid score of 0.85. And if you compare this to the different plant proteins, you can see that particularly the cereal-based proteins like oat, rice, or wheat are deficient in lysine and have an amino acid score of 0.5 to 0.35, depending on the type of protein. The maize protein here has an amino acid score of 0 because tryptophan is missing in this particular protein ingredient. And when we come to the legume-based protein ingredients, you can see here that they are, of course, for sure deficient in sulfur-containing amino acid, which is known from the start, and all the pulses are deficient in this amino acid. Then the next ones are the oil seeds, which are here represented by pumpkin, sunflower, and hemp seed proteins. They are also deficient in lysine comparable to what is present in the cereal proteins. And here on the right-hand side, you can see the potato protein, which is almost a complete plant protein, and the limiting amino acid is histidine. Whereas for almond, which is the limiting amino acid is lysine as well. So we, based on this, we were evaluating also the different single amino acids and the contents in the products. For example, for the sulfur-containing amino acid, where whey is significantly higher in those amino acids than the FAO reference. And what we found is that particularly rice is also having a higher content of sulfur-containing amino acids than, for example, whey or the FAO reference protein. Next, when we look at the lysine, as I said before, the whey protein has a higher content of lysine than all the other plant proteins, which means that we would need similar for the lysine. The lysine content is very high for the maize protein and even higher than for the whey protein. And therefore, if you compare the different slides I have shown now for the amino acid profile, you can see that when we use not only single ingredients, but if we use blends of different ingredients, we could complete the amino acid profile. And therefore, we started to combine different sources of those limiting amino acids. And if you look here, for example, on one of the examples we were determining, here on the right-hand side, you can again see the reference amino acid composition of the FAO protein compared to the rice protein. And the rice protein would have an amino acid score of 0.55 here, as the limiting amino acid is lysine in this protein ingredient. And if you look on the P protein, which is here on the right-hand side, which is limiting in the sulfur-containing amino acid, but high in lysine. You can see the P protein has an amino acid score of 0.91. And if you combine those two in a ratio of 28% of rice protein and 72% of pea protein, you can see that we got an amino acid score of 1.08, particularly as the lysine content is very high with 1.08. This means by combining different proteins, you could adjust the amino acid profile to what you want to have, even if you use plant proteins in comparison to animal proteins. But now the second goal was how to have attractive sensory properties of those protein ingredients. And again, we first of all determined the sensory profiles and the different sensory attributes of the different single plant protein ingredients, which can be shown here. I guide you through this PCA, of course. What you can see here on the F1 is that mainly the whey protein, almond protein, oat protein, chickpea, hemp, and soy protein have either milky vanilla-like and sweet tastes or almond-like nutty tastes. And those tastes or flavors are representative for those ingredients. And if you look on the other side of the F1 axis, which is displayed here on the left-hand side, you can see that all the other proteins are separated on this side. And what you could clearly see is that, for example, the potato protein revealed an algae-like, fishy, greeny taste, which was not so attractive to most of the consumers. And also the other different ingredients were characterized by bitter, pea-like, or greeny notes, so that the different ingredients should be combined to have attractive profiles. However, none of the plant proteins had similar taste or flavor components than whey had. And if we look now on the sensory profiling on itself, particularly on the maize protein and the potato protein, you can see here that, for example, the maize protein had a fecalic note, which is completely not acceptable to the consumer, and we have a certain bitterness and also a maize-like aroma in those protein ingredients. On the right-hand side, you can see the potato protein, which is clearly distinct by the algae-like fishy taste, which was also not acceptable to the consumers. And by including the older consumers, we needed to omit those protein ingredients in our developments, despite their high amino acid scores, which was displayed before. Now I wanted to go to the third goal, which was the in vitro digestibility of the proteins, which we did determine with an in vitro digestion method, where we applied pepsin, trypsin, and chymotrypsin as enzymes. Those enzymes cleaved the peptide bonds from proteins to peptide and amino acids, but some of the proteins which are indigestible remain as proteins. Afterwards, the amino acid residues are determined by a photometric method, adding ninhydrin as a reagent. So you can add you can measure the absorbance of those rumen purple, which is a addition product of the amino acid with the ninhydrin, to calculate the in vitro protein digestibility. And here you can see the quality of the data compared to a rat digestibility score, which is quite similar, that in rats, the digestibility of the proteins is quite similar to this in vitro assay. And for the in vitro digestibility, we also included all the different protein ingredients I mentioned before. And here you can see on the right-hand side, again, the cereals. In green, the different legumes. In lighter green, the oil seeds. And almond and potato are on the right-hand side. And what you can see here, of course, is that most of the protein ingredients have a very high in vitro protein digestibility, except for maize, which is significantly lower than all the other products. And based on these results, we were then going back again to our aims. So we had the 3 different objectives to combine our ingredients to have a plant protein blending which is optimal in the amino acid profiles, has a high sensory attractiveness, and has a high in vitro digestibility. And for this, we developed 6 different plant protein ingredient blends together with dietary fibers. We added to the protein blends always 20% of a pea fiber, which was the plant protein fiber we decided to go for. And for this, we developed 6 different plant protein blends, which could be used either in savory applications, which means like soups or something like that, or in sweet applications like porridges. And based on these, you can see here the different plant protein fiber ingredient products had different combinations of proteins in it to adjust for the optimal amino acid scores. And now, of course, the question is how those different plant proteins are perceived by the consumers. For this, we also did a sensory profiling. I'm sorry, it's written in German, but I explain shortly what you can see here. On the left-hand side, you can see the savory products, which is PP1 to PP3 with the different combinations. And what we experienced is, for example, that the combination of pea protein and rice, here displayed in blue, had a very sandy mouthfeel, and therefore it was not followed up to bring it to the application. On the other hand, the PP3, which was also a savory product combined from pea protein and pumpkin protein, had a very savory note and displayed an umami flavor, which might be nice if you add it to some of the products. On the right-hand side, you can see the same for the sweet PPFs, which were also characterized by the different attributes, and all the different PPF products were rated quite similar. However, the PP4, which is a combination of pea, soy, and oat protein, and the PP6, which is a combination of pea, soy, and rice protein, had a very high viscosity if you put it in your mouth. So they are generating viscosity due to the different protein ingredients. Therefore, we decided to move on further with those differences to determine the in vitro digestibility of the different protein blends. And here you can see, first of all, in blue, the protein blends alone, and then in the striped blue one, you can see the different protein, plant protein fiber products, where the fiber was added, and how this impacted the in vitro digestibility. You can see all the different products had a very high in vitro protein digestibility. However, if we added the dietary fiber, those in vitro digestibility was a little bit lower, but it was not that significantly different at all. And now, to summarize what we found out is we had developed 6 innovative plant protein fiber products with an optimized amino acid profile together with all the consumers. The sensory properties of 3 of the plant protein fiber products, namely the taste, odor impression, and mouthfeel, were rated as acceptable by all the consumers. We did an in vitro testing, which demonstrated a very good digestibility of the plant protein fiber products. And from this state on, we selected 3 different protein fiber blends for which were provided for the mechanistic investigation in work package 3, which were namely pea and pumpkin protein as a savory product. And from the sweet combinations, we selected pea, oat, and almond protein, as well as rice, pea, and soy protein. And there, I think Dominique will discuss the results of the work package 3. And for work package 4, we provided 2 different plans for the human intervention studies, which were both the marked in bold, the pea, oat, and almond protein combination, and the rice, pea, and soy protein combination in sufficient amount to be used for those intervention studies. And for this, I come to the end and would like to thank again Susanne for doing the great work within the FATI project and you for your attention. I'm happy to answer your questions.
Thank you, Stephanie, for this brief overview of a lot of work, which we started together at the beginning, and then Susanne stepped in and took over. There are indeed already questions. What kind of proteins were used, isolates or concentrates?
It were commercially available proteins, and the most were, so it was a combination of isolate and concentrates depending on what was available on the market.
Yeah. And another question regarding transferability of in vitro results to in vivo results. That's a reasonable question. And this is why we also examined in vivo digestibility in work package 3. We will hear the results the next talk.
Exactly. So this was also what I would refer to.
Yeah. And what is your take on the allergenic potential of certain plant proteins, especially pea protein? Might it be advisable to develop protein mixes without pea?
We decided that the allergenic potential of pea, as it is not on the allergenicity list of the European Union, we decided that pea would be acceptable in this advice, and also the older consumers often consume pea as it's in their meals. Therefore, we decided that for this study, pea is a great protein ingredient. However, if you want to have a mixture without pea, we could think of other sources, but it's quite challenging because pea protein is readily available on the market, whereas other sources, for example, sunflower or pumpkin, are only available by small producers at the moment.
Yeah, thank you. No other questions at the moment. One question I am often asked is what is the future now of these product developments? Of course, we've tested selected products now in our human studies, but are you still working on a further development or even market appearance of the products?
At the moment, we are waiting for the results, of course, of the work package 3 and 4. And depending on this, we are going to discuss this with certain producers who might be able to bring this to the market. However, it's always the question which kind of claims could be added to those different powders. And yeah, it's always, it's still a way to go to bring those products on the market.
Okay. Yeah, there is another question. I think we could take the time to answer it. It's about taste evaluation. Was it a suspension of the powder, and were the panelists elderly?
Yeah, we did two different evaluations. We mainly suspended the powders because in the real meals they are also suspended, and no one would consume powders as single ingredients. So we did the sensory evaluation in suspension, and we did it both with a trained panel, and we did it also together with elderly consumers, which was conducted at the FIU and at the other study centers. And then the results were combined to select the three most promising protein fiber ingredients to be used for the different studies.
Thank you again, Stephanie. Very great presentation and discussion also.